ever stumbled across the term glycogenolysis and are not completely familiar with it, then you're absolutely right here. Hello and welcome back to another video. Glycogenolysis is a metabolic pathway which generates glucose from stored glycogen. And by now you've already heard the term glycogen quite often, so let's talk about what glycogen is. It's a form of short-term energy storage and a multibranched polysaccharide of glucose. It's used to store energy not only in animals but also in bacteria and fungi. And here you can see a scheme of it. Actually, it's predominantly produced and stored in the liver, but also in skeletal muscle. A healthy liver can thereby store between 100 and 120 grams of glycogen. And well, when we're talking about skeletal muscle, it also, of course, varies between people, but it's approximately 400 grams for a 40 kilo adult. For the chemical structures, we have linear chains of glucose residues, and each chain gets formed by 8 to 12 glucose units. Those are either 1,4, alpha-1,4 or alpha-1,6 linked, which depends on the carbon atom, and here the examples for 1,4 linked ones and here for 1,6 linked ones. Now, we've already heard glycogenesis is a metabolic pathway that generates glucose from glycogen, which is the storage form of glucose. Now, the first enzyme is the Glycogen Phosphorylase, which can cleave the alpha-1,4 bonds. But as soon as we have an alpha-1,6 bond, we need a transferase and a alpha-1,6 glucosidase to continue with the degradation. And we can cleave multiple glucose molecules from one glycogen, which can contain up to 60,000 units of glucose. So this glycogen phosphorylase forms glucose 1-phosphate by cleaving the bond between the glycogen brand and a terminal glucose residue. So now glycogen 1-phosphate can be converted into glucose 6-phosphate by the so-called phosphoglucomutase, which is the same enzyme as in glycolysis. There's a separate video about glycolysis if you're not completely familiar with this term, which is linked above. Now, liver, kidneys and intestines can then convert this glucose 6-phosphate into glucose and secrete it into the blood. And we have to remove the phosphorylation so that the molecule can exit the cells. Muscles are also capable of taking up glucose 6-phosphate to make glycolysis and generate ATP, but in contrast to the liver, they do not distribute their glucose within the bloodstream, but rather use it for contraction. You can pause the video to take a look at the drawings and the respective enzymes for liver and skeletal muscles if you want to. So, but when do we use glycogenolysis? So it's activated when the ATP levels and so the energy levels are low. This also means that the blood glucose levels are low. Therefore, glycogenolysis is essential during fasting periods. And in general, it's essential for the fight or flight response and reacts to hormonal and neural signals. When we're talking about hormonal regulation, glucagon and insulin are really essential, whereby evidence indicates that insulin inhibits glycogenolysis, and on the contrary, glycogenolysis gets stimulated by epinephrine, which is adrenaline. This also prevents glycogenesis, which is the process whereby we build up glycogen from glucose. And we need this inhibition because the energy is needed at the moment and we mustn't store our glucose. As soon as the fight or flight response is over, glycogenolysis gets inhibited again, while glycogenesis can work again. So you might be a bit confused about the term glycogenesis and glycogenolysis. So glycogenesis is when glucogen gets formed from glucose. In glycogenolysis, we broke down those glycogen molecules to release glucose. So you mustn't confuse those terms. And there are also known pathologies associated with the ability to store glycogen, and whereby usually the enzyme function is impaired. Those disorders can be both acquired and genetic. Some forms of those are rather dramatic and others are less severe, and the treatment always as always depends on the cause. Now this was a quick overview of glycogenolysis. If you want to take a closer look at the enzymes, go back to the respective drawings, and otherwise I hope you enjoyed this video. If so, don't forget to give it a thumbs up and to subscribe to the channel to see weekly uploads. See you in the next video.